So, welcome to the second video on protein engineering. As a short recap, you saw that protein engineering can be done by computational assisted methods, basically using focused mutagenesis or by random mutagenesis methods. As a rule of thumb, when can we use computational assisted methods? We can basically use them for properties that you can term as localized in a protein structure. So you have an active site, and everything that is close to the active site has to do with activities, selectivity, substrate profile. These are properties that can be tackled in a straightforward manner with computational assisted methods. And also thermal resistance is quite well understood. However, there are methods that are not so well understood on a rational basis, and for this you can use random mutagenesis methods. This would be properties like organic solvent resistance, resistance against salt, pH stability, but of course you can use random mutagenesis methods also to improve activity, selectivity, or substrate profiles. There's one point where we should be very clear when we do protein engineering, how we are doing it. So all the mutations are basically done on the DNA level. So you basically mutate the DNA, and this is then translated on the protein level, as we saw, to an amino acid exchange. And what is quite interesting is that for every mutant that we obtain, we have only one variant in there. So every colony that grows on an agar plate at the end comes encoding the same mutant variant. Why is this the case? The reason for this is that the uptake of DNA as a large and negatively charged molecule is basically very inefficient by a cell. So in principle, only every 700 to 1000 cells gets a DNA molecule inside. And this DNA encodes an antibiotic resistance so that only those cells can grow that contain the gene, the mutated gene. So every colony that grows in a single agar plate is a single protein, encodes a single protein variant. So when we think about protein engineering, the first thing that we have to take into account is the protein sequence space. The protein sequence space gives us a feeling for the opportunities and challenge in protein engineering. So when we now look for a small peptide, a small protein that consists of 100 amino acids, nature offers us per amino acid position 20 different building blocks to exchange them. So that means that in a small protein we have 20 to the power of 100. This is this large number of different protein variants that can theoretically be generated by natural design. And when we now think about engineering proteins and we screen 1 million variants of the proteins that we have generated, the fraction that we cover there in terms of the protein sequence space is, as you can see, with all the zeros, and I'm not talking slow enough, not really significant, and I hope you can agree to that. So let us put this number in a perspective. So when we now look at all the stars that are in the universe with 10 up to 10 to the power of 24, and you look at the number of the protein sequence space, and you would multiply this still with billions, you can still generate theoretically more protein variants. This, on the one hand, is a very positive thing, because there should be no reasons why when we use enzymes that they should not be stable in our chemical processes, work in organic solvents, tolerate high amounts of substrates or products. On the other hand, on the negative side, we have to find a way to navigate efficiently through the protein sequence space, because we have to admit that we will never be able to generate all this 10 to the power of 224 variants and to experimentally test them. It's simply not possible, and we have to accept it. So that means that we need strategies to cover how we navigate through the protein sequence space. So now where is the upper limit in terms of screening? This we will basically discuss then in the third lecture. Let's start first with directed protein evolution. So I'm very happy that this year the Nobel Prize in Chemistry was awarded to my former postdoc supervisor Francis Arnold for the technology of directed enzyme evolution. So why is this the case? So what can directed evolution do? Directed evolution starts with one gene that encodes the one protein variant. So in the first step, and that's the first key technology, random mutations are generated in the gene, which in 2/3 of the cases result in amino acid substitutions on the protein level. And then you need a good screening or selection system to select for the few variants that are improved in the property you're interested in. So the cool thing about directed evolution is that every property that you can in principle reflect in a screening system, you can select your protein variants for it. Then you isolate the corresponding gene and you start this process iteratively again until you have improved your catalyst as far as you would like to have improved the catalyst. And another key feature of directed evolution is you don't need any hypothesis. You don't need any pre-knowledge. As long as you have a screening system, you can improve the catalyst you are interested in. And when you then analyze the substitutions, you can on the molecular level understand fundamental principles how to re-engineer, to reprogram your enzyme to non-natural conditions. So how are the key features and the key parameters when we look at a standard directed evolution approach? Now let's look on the key performance parameters of directed protein evolution. So a standard or traditional directed evolution experiment starts with a mutation frequency of 1 to 5 mutations in 1,000 base pairs. This leads to approximately 1 to 3 amino acid exchanges. Usually you screen only a small amount of clones. And when you take the numbers from before into account, is it not quite surprising that by screening a few thousand clones, you find improved enzyme variants? But this is basically what is happening. And per round of a whole evolution campaign, you usually get an improvement of a factor in activity of 1.5 to 2.5, so 2 on average. This means if you want to make a good catalyst, first round twofold improvement, 4, 8, 16, now it starts to get interesting, 32. You need iterative rounds of directed evolution. And then you end up in your protein with 6, 8, 10 amino acid substitutions. And when you then analyze them, very often there are ideas what the positions do, but it's not clear what the individual positions really have an effect. And sometimes you have positions in there because you get 1 to 3 amino acid exchanges that do not really contribute. And these iterative cycles, they cost time. So if you have an experienced person, he or she can perform the evolution campaign in a year. if you have a person that has to get trained, a PhD fellow, then it might take 1.5 years until you really have good results. And when we now look historically until 1994, there were less than 14 successful directed evolution campaigns in Web of Science. This went up to 200-300 in 2003, 2004. And since 2010, it's approximately 450 to 550 successful directed evolution campaigns per year. But it's not growing anymore. And there are reasons, methodological reasons for that are related to the time requirement. So essentially, directed evolution enhances, as a summary, the natural evolution millionfold, so that you can reprogram, re-engineer your enzymes to the needs that we have in a sustainable bioeconomy. So let's have a look to the key technologies that are there. The first step was the diversity generation. These challenges are technically solved. So the standard methods to do random mutant libraries is error-prone PCR. This is essentially a PCR reaction. As you know, you have a gene, you heat it up, the strands melt, one strand is there, and then you have the polymerase, primer binds that the polymerase elongates, and instead of magnesia, you add additional manganese ions. And these cause that the polymerase makes more errors. So you can see, for example, here in this gene, and then the full-length gene gets synthesized. So on average, that's 3 to 5 mutations per 1,000 base pair, depending on the enzyme class that are used in traditional evolutionary experiments. And when you think that such a PCR reaction takes 2 to 3 hours, you can generate, like in a standard PCR amplification, easily a huge amount of mutations and mutant variants on the protein level in a single PCR reaction. This is for random mutagenesis. For focused mutagenesis, there are also many methods available, and one method is the OmniChange method. It's in principle like you saw before, only that you do 4 or 5 PCR reactions. You do the assembly of the genes a little bit different, but you can still generate in one afternoon, when you tackle 5 positions in a focused manner, 3.2 million variants if the PCR works. So technically this challenge is solved. Now we can ask ourselves, when we look at a random library, an error-prone library, what do we find there experimentally? We can do this theoretically, and for example, computational with the mutagenesis assistant program, so the MAP program, you put in your error-prone conditions, and then it gives you for the protein this scheme here. Here you have all the amino acids that are in the protein. This is a stop codon. Here are the substitutions that you can expect in your error-prone library. And what you should strike your attention are these huge white areas. For example, here. And these are amino acid substitutions that are not obtainable with error-prone PCR. So despite this quite low diversity, error-prone PCR is still very successful in finding improved variants. And this MAP program was basically used to develop a benchmarking system to compare random mutagenesis methods for the first time on the protein level, and to set benchmarks in terms of developing mutagenesis methods to increase the diversity. But we can also experimentally look at the diversity that is generated. And when we now take a small lipase gene and we sequence there from an error-prone library 1,000 mutations, this is like screening a small library. We can do this with a high mutagenesis frequency, 11.7 base pair mutations per 1,000 base pairs, and sequence 1,000 1000 variants, then this is what you totally get as a result. These are all the amino acid substitutions that occur in this library. So you have positions where you have no substitution, 1, 2, 3, and sometimes even 4. So on average, you have a little bit more than 2 substitutions per amino acid positions. And remember, nature offers you 20 building blocks, so 19 other amino acids compared to the amino acid that is present. So 2 out of 19 is not bad, but also not that impressive. And if you now screen such a library for an improved resistance, for example, against a certain salt, it's a completely non-natural conditions, you can, by this approach, find 15 or 18 positions that give you an improvement in the resistance against the salt. So improvement means after taking account the standard deviation, every clone that has 3 times higher activity than beyond the standard deviation is accounted as a positive one. So 15 to 18 positions shows you can improve your catalyst. More interestingly is what kind of improvement is obtainable if you would be able to sample through the whole natural diversity. So you can do that with the same lipase. You make all the 20 amino acid exchanges at each position, which is a very extensive work, which takes 2 co-workers up to approximately 2 years to do that. And then you can look there at the amino acid exchanges with the same property. 100 amino acids. So how many positions do they really contribute to an improvement? And the result, when you look at this, is quite astonishing. So more than half of the positions, lipase has 181 amino acids, and more than 100 positions contribute. So more than half of the positions have at least one amino acid exchange that gives an improvement. So what does this now mean for directed evolution? This means that when you use error-prone libraries, approximately you only find 20% of the positions that really contribute. You can say, oh, who cares about salt resistance? But this is quite similar for organic solvents, for detergents, and many other properties. So you cannot deduct from error-prone experiments conclusions to general principles of re-engineering proteins. So if you want to do that, you need these libraries with the full diversity. And this is very important. So if we want to understand the principles how to re-engineer proteins, we need this full diversity libraries so that we can more and more use computational methods to improve our catalysts. Now you can ask yourself, what are the molecular reasons in error-prone PCR for this low diversity? And the molecular reasons are threefold. In the first case, you use a polymerase to amplify the DNA. And if you make 100 mutations, in 63 cases you get a mutation from A to G or from T to C. So the polymerase has a preference to replace a big base by a big base and a small base by a small base. And this limits on the protein level the diversity. If you go now for a traditional directed evolution experiment and a mutation frequency of 3 to 5 mutations in 1,000 base pairs, it's statistically very seldom that 2 mutations are next to each other. But you need 3 subsequent nucleotides to encode one amino acid, one building block for a protein. But if you don't have mutations next to each other, you only hit one nucleotide in this so-called codon. And as you can see here, this further limits the diversity that is generated. And what we should not underestimate and actually be quite grateful for is the organization of the genetic code. Because with this mutation, the preferences from polymerases also inside us for this change big to big, small to small base. And the disorganization of the genetic code will limit on the protein level the chemical diversity that is generated. In the meantime, in the last couple of 10 years, many other methods were developed to improve the diversity. So here you see error-prone PCR. Here you see one method termed CSAM, sequence saturation mutagenesis. It was commercialized. And the actual CSAM method. And I hope you can agree that this part here is a little bit greener than this part. So there the problems are technically more or less solved. But these findings lead us to the next question because of the diversity. How can we explore the protein sequence in a time-efficient manner and maximize the obtainable improvements? You don't want all the time to make a library with the full diversity. This takes way too much time. And there are several strategies to balance the throughput and the screening efforts in order to get to a very good catalyst. And one method that that I would like to emphasize, to advertise for a little bit, is basically the Novolution method, which limits experimental efforts and generates a molecular understanding of each position that was targeted. Novolution has 4 phases. In the first phase, we do a standard error-prone or other mutant library, do some screening, and identify approximately 14 to 16 positions that potentially contribute to the improvement we're interested in. Sometimes you have 1 amino acid exchange only, sometimes you have 2, sometimes you have 3. So in the second phase, you target each of these positions and you generate all the natural diversity with all the 20 amino acids. And usually if you found, for example, in phase 1, 12 good positions, after checking each of the positions, only 8 or 9 really contribute. And very often, since you know from before that on average you have only tested 2 amino acids on the position that you found, that when you check now all 19s, you find even a better improved variant. And if you then do the sequencing, then you know after phase 2 the positions that contribute. And you know for each position you have the amino acid exchange, you know the chemical nature of your amino acid exchanges. So you basically know whether it's the size, the charge, the H bonds that really contribute to your property improvement. You generate knowledge. And if you then now analyze on the protein structure, basically where these positions are located, they usually cluster in 1, 2, or 3 areas that are quite close to each other. And if you then group them according to their location in space, you can then, for example, when you have 5 positions on one side, simultaneously exchange all the amino acid side chains. So you can have cooperative effects, for example, with a method like OmniChange that really boosts then the activity. If the positions are far away, you can individually recombine them in phase 4. And by doing this, you can very rapidly, with minimal experimental efforts, find really good catalysts. So you are now an expert in diversity generation by directed evolution. In the next video, we will then take a look to the high throughput screening methods and how high throughput screening methods matter in order to find very strongly improved catalysts. So thank you for your attention.